Shailee Jain speaking. You're a postdoc in Eddie Chang's lab at Berkeley. And I've actually followed her work. She's done fantastic work on decoding language in the brain and yeah, basically modeling language in the brain using LLMs and so forth. So looking forward to hearing more.
I hope it's not too loud. Hi everybody, I'm Shailee. Firstly, thank you to the organizers for including me in this fantastic workshop, and to all of you for being here this early. So today I'm going to be talking about some of my PhD work that I finished about 1 year ago, and my more recent postdoc work in the past year, all of which is under this really broad theme of trying to understand how the human brain processes language through the lens of really powerful artificial language systems. Now, before I begin, I just want to note that many of the talks you might have heard at this workshop, or really any talk you might have heard in today's day and age, probably begins with and centers around this idea that language models are extremely powerful, and somehow we now have these systems that are extremely good at generating language and generating language to prompts that we never thought they would be able to understand. But I want to, instead of telling you how good these models are, because I'm sure all of you already know, I want to actually overly simplify and think about how we got here. So as a vast oversimplification and ignoring the fact that NVIDIA just has amazing GPUs that we can use, the reason we have these powerful foundation models is, I think, based on two things. First, our ability to have really vast, large-scale, decent quality data sets. For example, any large language model is trained on the entire internet. So we have access to these huge data sets that these machine learning systems can get patterns out of. And we also have really, really good models now in a way that we didn't have before. So the transformer, for example, really revolutionized the types of artificial systems that we're able to build. And this combination of data and models has somehow gotten us to this point where we have things like Claude and ChatGPT and DeepSeek and what have you. But instead of talking about this revolution of AI, I also want to bring to your attention another revolution that is much more nascent, but I think is happening in parallel. And this is a similar data and model revolution in language neuroscience. So let's just start with data. Let me show you a few examples of the types of neural measurements we're able to collect today in ways that we haven't been able to in the past, and the amount or the scale at which we're able to collect these neural measurements. This first example, or this animation, is actually that of a 3D brain that is responding to somebody listening to a story. And what I'm showing you is the different amounts of blood oxygenation levels around the entire brain when our participant listens to a story. And I collected this data using functional MRI. So this is our first type of neural measurement, blood oxygenation level. And what this gives you is this very slow varying signal. You get information every 2 seconds about, but you can map the entire brain. And just to convince you that we're able to collect something like this at scale, I want to talk to you about this large-scale data set that I collected in my PhD work that was over 20 hours of data for every human participant. Another type of data modality that I want to bring to your attention is electrocorticography. So while functional MRI is really slow, you can also place these electrodes on the surface of somebody's cerebral cortex or across the depth of their brain. I apologize for not giving you a trigger warning about an actual brain there. But what these electrodes can then do is measure activity across hundreds of thousands of neurons at a very fast temporal scale. So this is another type of neural measurement. A third type of neural measurement is this really amazing technology that we've only been able to leverage in human neuroscience in the past 2 or 3 years. You might have heard about this in Laura Gwilliam's talk on Tuesday. And these are neuropixels probes. I'm sure from this distance you're not able to see that little red wire, but that neuropixel probe goes across the depth of somebody's, of an awake human's cortex. And through that single wire, we're able to record activity from thousands of single neurons in this person's brain. So for the first time, we have access to not only these really slow but whole brain signals like fMRI or really fast temporal population signals like electrocorticography, but we're able to capture signals from single neurons in an awake human's brain while that participant is engaging in language and communication. So I just want to, just because how exciting that data is, I just want to quickly show you what it looks like. On the x-axis at the bottom is time. On the y-axis are 1,200 random neurons that we've picked out. And what you're going to see on the top is the auditory waveform of our human participants listening to speech. And I'm going to show you the spiking activity of these 1,200 neurons in different colors. And at the bottom, I'll sum up the activity across all the neurons to show you what the population activity looks like.
Mollusks are a case in point.
Does this bother you?
A flame would use up air.
12 o'clock level.
So what I hope you can appreciate from this really quick video are two things. First, you can notice that these neurons have very, very different firing patterns. There are some neurons that fire very little, and there are some neurons that fire all the time. But the second thing you might be able to appreciate is that these neurons are also time-locked to speech. So when speech happens, the population activity rises. When there's silence, it goes down, and then it goes up again. And so what I'm trying to demonstrate here is that neuroscience has gotten really, really good at measuring all of these different types of neural signals that operate at different spatio-temporal scales and can tell us very, very different things about how the human brain is able to achieve a feat like language. And not only do we have diverse types of data, we also have lots of data. So at the beginning, I told you about the fMRI data set that I had collected with others in my PhD lab. We've open sourced all of this data as an aside. I've also written some helper code if you want to build the models I'm going to discuss today. So please check it out. But aside from getting 20 hours of fMRI data per participant in our electrocorticography work in the Chang lab at UCSF, we've also collected over 15 years data from over 300 human participants listening to more than 1,000 hours of speech. So you can already appreciate the types of scale that neuroscience is able to collect in a way that we never had before. And coming to the NeuroPixels work, we have data from around 100 patients and 20,000 single neurons in these patients. So you can already see that in each of these modalities, we have so much data and so much to learn about how the brain is processing language. But now that we've solved the data question, or we're pretty confident in the types of data we've collected, how do you actually build models of the brain? Fundamentally, what we want to know is how does the brain go from the speech that somebody hears?
Good question.
Yes.
The plot that you just showed.
Yes.
Where do the responses of the neurons start? Because you showed like it started at 2200 or something. Just the last, yeah, that one, yeah.
2200. Oh, sorry, that's the number of neurons. I'm sorry. So each line here is a different neuron.
Yes.
Yeah.
But you said you said you had 1200 to begin with, no?
Oh, sorry. I was just plotting 1200 as an example, but we have access to about 20,000 neurons. Okay. Yeah, sorry. So across all of the data we've collected, we have over 20,000 speech or language responsive neurons in the human brain. But I've subsampled 1200 here for demonstration. OK, so now that we have all of this great data, we want to build models. And so fundamentally, the question that we're trying to answer is how does the brain go from the speech that somebody hears to the brain activity you're recording? Here I'm showing you activity from two different parts of the brain. Those little cubes are known as voxels or volumetric pixels. But this could be anything. It could be single neurons. It could be MEG sensors, ECOG electrodes, what have you. But ultimately, our model needs to learn a mapping from the speech that somebody heard to the brain responses that we recorded. So in a very, very simplistic sense, what language neuroscience typically did when I first started my PhD was to solve it like a simple machine learning problem. The first thing you do is you think about the types of features that the brain could care about. You build your feature space. For example, I could represent every single word in our story with its word embedding. I bet none of you thought I was going to talk about glove or word2vec, but you can do that. You can represent every single word here with a word embedding, and then you can learn a simple linear regression to go from those word embeddings to the brain responses that you recorded. This is a very simple setup, but it works well, and it allows you to test different types of feature spaces. And in a very classical machine learning sense, you then give it a new story the model has never seen before, and you test your model. Now, this is great, and this was really the bread and butter of computational language neuroscience. This was the best model we had when I first started my PhD, but there are obvious concerns here. To begin with, we all know that word embeddings are static, and language is highly contextual. So when I first started my PhD, I wanted to find a way to just improve these computational models. How do you incorporate, for example, context in these models? How do you build a model that can predict brain activity not just based on a single word, but everything that has happened before? And so my simple idea at the beginning was, I'm just going to use a deep neural network, because why not? Elmo had just come out that time, and Elmo was an LSTM that worked really well. And so we figured for every single word in the story, don't use just that word, use the preceding context, put it into a language model. Again, it doesn't matter. You can choose your favorite language model. But then you can extract these contextual hidden state representations from these language models, and you can just follow the same linear regression pipeline. And this actually works incredibly well. I'm about to show you a plot from my first paper, but at this moment, with powerful models like ChatGPT or whatever, we've basically reached noise ceiling in terms of how well we can predict any part of the brain that cares about language. These models are extremely good at predicting brain activity. And this was my first paper. I know this sounds like a very simple idea, but it was really exciting at the time, because this was the first paper ever to show that you can use language models to predict fMRI activity. Before this, Tom Mitchell and Leila Vehipe had done really, and Alana, who's here, had done really exciting work on a similar idea for MEG, but we wanted to do this for fMRI, and we wanted to infer different functional properties of the brain using this model that I'm not going to talk about today. But let me just quickly show you how good this model works. Again, this is from 2018, so take it with a grain of salt. What I'm showing you is a flattened out brain surface. I took the left and right hemisphere, and I just flattened it out. Anything that is dark here means that it does not care about language. It does not respond to language. Anything that is bright, especially if it's bright white or red, implies that these language model-based representations helped a lot. And you can already see, even with this now ancient LSTM model, it works really well. You can predict a lot of the brain really, really well. So what this gave us for the first time is very powerful predictive models of language in the human brain. Now, this is great. We've now built a predictive model. It does really well. It has great generalization and test set performance. But a rigorous scientist might ask, what's the point, right? Like, did I, in fact, trick you into believing that this is a good idea by just replacing one thing we don't understand, which is the human brain, with something else we don't understand, which is the deep neural network? I would like to argue, and in the rest of my talk, I'm going to try to convince you that this is, in fact, a good idea, because we can interpret these models, we can interpret language models and their computational models in ways you can't the human brain. You can break these models apart, you can do ablations, you can do many interesting generalization tests, and hopefully this can tell us something new about the brain that is simply not possible given the time and like any experimental constraints that we have, and also the fact that you just simply can't go into a human brain and knock out whatever you want. So in the rest of the talk, I'm going to present 4 different ideas that I've developed, both in my PhD and in my postdoc work, to interpret the human brain using language models. And I'm going to go into each of these ideas in detail, but first let me just tell you what they are. The first idea really focuses on the fact that we have a very, very good predictive model. So given that these language models are excellent at generalizing and at producing brain activity independent of what the language stimulus is, we can actually use these models to do in silico experiments. What I mean by that is, instead of painstakingly collecting lots of different brain data for all the different hypotheses you can test, you can simply just use your amazing generative model to tell you what the brain responses might look like in different stimulus conditions and help you narrow down the types of hypotheses you might have about what the brain is doing. So you can replace your in vivo expensive experiments by doing in silico experiments using a powerful predictive model of the brain. So that's idea 1. Idea 2 focuses on the deep neural network or your language model itself. in this particular idea, we thought, why not just design interpretable language models from the ground up? If we design an architecture or a system that is interpretable, perhaps we can extend that interpretability to the predictive model of the brain, and we can understand something about the brain. So the second idea is designing interpretable LLMs from the ground up. The third idea also focuses on interpretability, but instead of devising new architectures, which can be very difficult, we can also use different computational components of existing architectures that are interpretable. So just to give you a sneak peek of this section, positional encodings and transformers, for example, serve a very specific purpose, and they're very interpretable. And perhaps by using those models, we can understand something, or using those feature spaces, we can understand something about how position is encoded in the brain. Or the attention heads, for example, for transformers are extremely interpretable, as a lot of amazing people have shown in AI. And again, you can use the interpretability of an attention head to understand what a brain region that is well predicted by that attention head might be doing. And the last idea is this is a little out there. I'm not going to show you actually any results because this is work I'm doing at the moment, but I'm just very excited about it, and so I wanted to talk about it and maybe get all of your feedback. So you might have noticed that all of the models I'm describing, even throughout my first 3 sections, will follow this linearization paradigm where you extract some features, which might be non-linear, but then you learn a linear mapping from those features to the predicted brain activity. And this works really well, especially in low data regimes. But given that we have all of this vast neural data now that we never had before, my last grand idea is to build a silicon brain. And I know that phrase is confusing and ambiguous, but what I'm basically proposing to do is why not build an end-to-end system or a foundation model of neural activity that doesn't have this feature extraction step, but you basically put in the language somebody heard, train an entire artificial neural network by asking that model to directly predict the brain activity. And if you have this artificial network that's trained on the brain data directly, you can use any of the interesting AI interpretability ideas that people have developed to now inspect the inner workings of that model and understand something about the brain.
I have a question a bit more about this model. It's trained on multiple different participants, correct? Yeah. So how much variation is there between, let's say, me and the people around me in our exact brain activations? If you were to do this, would it make more sense to train on a specific brain or a specific population of people? Like, how important is that when you're creating this model?
That's actually a great question because there's 2 parts to it. One is a lot of our brains are very, very similar. In fact, Ev, who's here, has done really beautiful work to show that independent of the language you're hearing or any demographic information, people have very reliable parts of their brain that light up when they're listening to language. So we all process language very, very similarly. But there is this small consideration that we all have very different brains. Our brains look different, they wire different, we have different experiences. And so there are obvious subtle differences in how an individual processes language, even though on average we're all doing the same thing. But this idea of studying individual differences has been very elusive in language neuroscience just because it's a very small effect size and it's really hard to actually know how to study individual differences. But I'm not going to go into the details of the model I'm actually building, but I can just tell you that what I'm currently working on and proposing to do is have a model that's trained on multiple people, but have, say, an embedding space that represents any unique participant. And this allows you to not only study individual differences by inspecting that participant embedding space, but also capture a more generic foundation model of language in the brain. So we want to be able to do both of these things in tandem.
So I have a question regarding between listening and reading. For a neural network, it doesn't make any difference. It's the same thing, but for human, isn't the patterns different?
Yes, again, a great question. So listening and reading are quite different depending on where you're looking. So listening, for example, activates a lot of lower level primary parts of your auditory cortex, which is around here. But there's also parts of the auditory cortex and beyond that are activated jointly by reading and listening both. So this is an oversimplification. Listening activates primary auditory regions. Reading activates primary visual regions at the back of your brain. But after that, after all the primary processing is done, a lot of the higher level stuff like semantics or syntax is processed by the same brain regions. It's still a work in progress. We're still trying to understand exactly how this works, but that's the idea. Okay. So okay. So let's get into it. Let me talk to you about the first idea, which is in silico experimentation. So let's go back to that model I first showed you. So in this model, we're using these contextual representations of every single word in our story, and we're using linear regression to predict the activity of these different voxels in the brain. But in this particular study, I was interested in figuring out what that context actually means for any specific brain region. Specifically, I wanted to know if different parts of the brain respond to different types of semantic concepts. Like, for example, is there a part of your brain that really lights up when there's emotional content, and a different part of your brain that really lights up when somebody says something about numbers or colors? But I also wanted to ask the secondary context question. Are there also parts of your brain that respond to different amounts of temporal information? Like if somebody says the word happy versus saying, I'm having a wonderful day. Are these processed similarly? Is there some relationship between semantics and the amount of information that a brain region needs to integrate to arrive at that semantic concept? And to study this question, I developed this in silico experimentation paradigm. So first, let's just focus on semantics. Let's forget about timescales for a second. Now, what I did was I first just constructed a gigantic corpus of phrases that had very different semantics, very different syntax, different word frequencies, what have you. This was just a 100,000 phrase corpus. And if I had infinite time and money, what I could basically do is make each of my participants listen to each of these individual phrases in some natural context, and then evaluate if every single brain region likes some phrases more than others. If a brain region likes some phrases more than others, I could then evaluate, are those phrases semantically coherent? Because that would tell me that that brain region likes a specific semantic concept. But I don't have infinite time and money, so I went to my trusty predictive models of the brain. And I simply simulated brain activity by using my language model-based predictive model of the brain. So for every single brain region, I simulated what its activity would look like for these 100,000 phrases. I could then rank these 100,000 phrases for every brain region, and then I could evaluate whether the top phrases that a particular voxel likes are semantically coherent. So let me give you a quick example of what this looks like. Yes, yeah. So the model that I used basically takes every single language stimulus, puts it into a language model. It could be GPT or an LSTM, or it doesn't really matter. It puts it into a powerful language model, and then it takes the hidden states of those language models to then do a linear regression from the hidden state to the brain activity. And this works really, really well. Amazing test set performance. We did a lot of generalization tests that I'm not showing here, but it works really well. And so you can be fairly confident that for many parts of the brain, this is a reasonable in silico setup to use. Okay. So for this particular brain region, so not every part of the brain loves semantics, right? Like that would be a bit weird. But there are parts of the brain that do. And this is one example. So this particular part of the brain, for example, really loves phrases that capture this concept of politeness. So for example, this part of the brain loves phrases that say things like, thank you for the safety and delivery of my daughter. I would have thought of this as kind, the integrity to tell me. It's really great to have you, etc. So it really responds strongly to this concept of politeness. And it also happens to be in the prefrontal cortex, which is not only responsive to language, but is involved in many, many different types of processes in the human brain, like decision-making and emotion regulation and so on and so forth. This is another, yeah. Yeah, so these are 2 millimeter isotropic voxels.
And so this politeness region, is it isolated to 1 2 millimeter voxel, or does it extend over a few neighboring ones?
Great question. This is 3 slides down, but you actually find these like cohesive patches. So it's not just a single voxel, but it's like a couple of voxels that are cohesively responding to a similar concept. It's a little hard to see here, but if any of these brain regions have similar colors, they respond to the same types of phrases. Yeah. So the second voxel really responds to this concept of hand and mouth actions. So every time there's a phrase that has some hand and mouth action, like split into a wide grin, shook my head and nodded, she could laugh, hold her hand or kiss her, this voxel gets very excited. And so this is nice. We're able to use this in silico paradigm to estimate the types of semantic selectivity that different brain regions could have. And the next thing I want to do is now to go in and check how the context works for every single phrase that a particular voxel likes. Yes?
Yeah, I was wondering, like there are clearly a lot of different sections of the brain that you could be looking at it. Were you just randomly choosing some and then identifying some patterns, or did you have some quantitative method to identify the sort of cohesive similarity, or did you have a hypothesis beforehand about specific areas?
Okay, again, a great question. So my voxels are 2 millimeter isotropic, right? But for participants who have great brain activity, like just really good signal to noise ratio, about a third or more of their brain reliably responds to language and is well predicted by my model. Now, not all of these voxels care about semantics, but a huge percentage of them do. And so I run into this issue where it's very, as a human, I cannot go in and figure that out. So I used a bunch of different strategies. My first strategy was I used like a BERT score to identify how cohesive the phrases are. I'm not showing you what my significance procedure is, but I had a involved bootstrapping procedure to decide whether a phrase was significantly chosen by a voxel or not. And then I used BERT score to evaluate cohesiveness, blah, blah. And then I did this. I tried it many different ways. I tried like an n-gram-based approach to summarize the topic, etc. But then I took the lazy approach of feeding all of my phrases into ChatGPT and asking it to summarize the topic of those phrases. So once I'd use an NLP method to establish that there was some semantic coherency between the phrases, the topic summarization I did using ChatGPT or an n-gram-based topic model and things like that. Cool. Okay, so now we've identified some parts of the brain that care about semantic concepts. We now want to look at how much of time they need to integrate to arrive at that concept. So to do this, I developed another in silico experiment. Here, for example, is a phrase that both the teal and purple voxels like a lot. Now, I want to understand which words in this phrase are contributing to generating the response of each of these different voxels. To do that, I can do this really simple experiment wherein I knock out one particular word. For example, I knock out the word laughing. I feed it into my language model, and then I evaluate how much the predicted response changed. Now, in NLP terms, what this means is I can replace the embedding of the word laughing by a random embedding, a noisy embedding, or a zero embedding. It doesn't matter. You get the same results. But I'm basically trying to understand how much that word contributed to the hidden state of the language model that ultimately predicted that voxel's activity. And what you find is these very interesting and different patterns. On the x-axis are the individual words I knocked down. On the y-axis is the percentage drop in the voxel's response when that word was knocked down. And what you can see is even though these two voxels like the same phrase, they're picking up very different types of information. The purple voxel, for example, is combining information over the subphrase laughing a little, but the teal voxel sort of cares about the subphrase started laughing. And I'm skipping over a million steps here. I can talk to you about it offline if you're interested. But we then did a lot of analyses to combine this semantic selectivity and integration timescale idea together to understand what is the cortical organization of semantics and time jointly. I'm going to give you a very like a 30-second summary of our story in the interest of time. But what we essentially found is this. In this particular part of the brain, for example, similar to Surya's question, you actually find that all of the green voxels here care about the same semantic concept. So they all respond to numeric information that is conveyed through one or two words, so things like 3 or 9. But the neighboring voxels right here, all of the red voxels, care about time information. They care about the concept of passage of time, but spread across 2 to 3 words. So like the next day, the next morning, etc. Similarly, in a different part of the brain, the green voxels, again, are very coherent, both in their semantic selectivity and the amount of time they integrate over, And the red voxels are very coherent in the semantic selectivity and the amount of time they integrate over. But what we essentially found is some shared hierarchical organization, which is that this part of the brain, or this part of the brain, always has shorter time scales compared to this part, independent of what the semantics is. So there's this very complex relationship between the anatomy, the semantic selectivity, and the time scale of every brain region. And we were only able to arrive at this story by doing our in silico experiment. Sorry, just 1 second. But I do think that in this in silico paradigm, there's this obvious question, which is, how can we be sure that any of our estimations are correct? Like, our models have great R-square values, but how can we really trust them? So I did some follow-up work with Bin Yu's lab at Berkeley and Microsoft Research to actually devise this closed-loop experiment wherein you generate the types of stimuli or the types of semantic concepts that you think are going to maximally drive a brain region, and then you evaluate in your fMRI experiment whether that is actually true. So we devised this closed-loop experiment in a follow-up study to prove that the sorts of functional properties I'm showing you here are in fact true, and in fact that is what you find when you do an actual brain experiment. Sorry, did you have a question?
Yeah, that was a question if you did that.
Okay, great. Okay, cool. So I'm going to skip ahead unless anybody has, yeah.
The different dynamics between brain areas?
Great question. So fMRI is very slow. You get an image every 2 seconds or so. And after this section, all of my work is going to be non-fMRI, and you might appreciate what having high temporal resolution can give you. But for that experiment here, for the experiment I was just showing you, we were looking at semantics, which tends to be quite slow. And we did a lot of control analysis to show that the sorts of ablation experiments we were designing were able to get at the functional estimates of that voxel. So if you change your acquisition rate or whatever, you don't really change the how the ablation mechanism works. Does that make sense? Okay.
So some of these semantic categories are very specific, right?
Yeah.
So how many of them are there, and are they discrete, or do they overlap a lot? I mean, I have no idea how big this space is.
That's a great question. So a lot of the heavy lifting to answer that question was done by my PhD advisor, Alex Huth. But I continued that work, and what we basically found is that it's definitely a continuous space. It's a very broad space. So you can roughly summarize the types of concepts you find into like 10 clusters or something. Like there's emotion, there's time, there's objects, numbers, things like that. But it's a very continuous space. So what I didn't show you here is you often get voxels that care about a mix of combinations or care about things like politeness or hand and mouth actions. So there's a lot of specificity, but it's definitely a continuous space. And it's a very broad space. Okay. Cool. So moving on to our next section, this one is really short. So this next idea, I've showed you how to do in silico experiments, but another thing you can do to interpret the brain is design interpretable language models from the ground up. And in this particular, sorry.
What's your takeaway from that? What is it more information about organization or why things are the way they are?
Yes. So our grand story of like the organization of the cerebral cortex in terms of these functional properties was that there are different brain regions that care about different semantic concepts, and the semantic concept can skew the type of timescales you observe. So what I didn't tell you is a part of the brain that cares about a concrete concept, like color, for example, tends to have a shorter time scale than a part of the brain that cares about something more abstract like emotion. So we find this very interesting relationship between how a concept is conveyed in language and the types of timescales we're observing in the brain. But at the same time, anatomy matters. So even if you care about time, if you're in this part of the brain, you care about shorter timescales as compared to this. And so we find this really nice hierarchy of timescales independent of what the semantics is across the brain. And we also find that you can't really identify any functional property in isolation. You need you need to know the anatomical location and the semantic selectivity of a brain region to figure out what the time scale can be. So there's this very complex three-way relationship between what those brain regions are doing. In terms of your second question about why it might be, I have hypotheses that I would love to explore in the future, but these are just hypotheses. So one thing that we've observed is a lot of the brain regions, like this part and this part, that tend to have these really long time scales, tend to also do things much beyond language. So they not only like language, they like a lot of different modalities, like vision, for example, watching a movie, or decision making, or all of these different processes. So these long time scale regions tend to do multimodal things. And what is interesting is that this hierarchy that we are seeing from this part of the brain to here, or here to here, is preserved in non-human primates as well. So if you look at a macaque brain, a macaque doesn't understand language, but it has this fundamental hierarchical timescale organization that we're observing in the human brain for language. And so I think there's a very interesting I think there's a lot of scope there to explore how musical processing or sequence processing in non-human primates could have given rise to the patterns that we see in the human brain for something like language, which is a sequential statistical problem as well. So moving on to the next section, here I want to talk to you about another interpretability technique, which is designing interpretable language models from the ground up. And to do this, in this particular study, we turned to our trusty friends, which are recurrent neural networks, just because of how interpretable they are. And I don't want to tell you what a language model looks like if it's an RNN. It's the same idea. But you basically take a standard recurrent neural network-based language model. But to make this RNN interpretable, what we did in this particular project is we tweaked the forget gate biases of each unit in this RNN. So we took every single hidden state unit in this RNN, and we fixed its forget gate bias such that we could control the rate at which that unit forgot information. And through this, we were able to build this interpretable multi-timescale RNN. For example, the yellow unit had a low forget gate bias such that it forgot information very quickly, so it just bounced around and had no history of what had happened before. But the purple unit, for example, had a forget gate bias such that it integrated information over long spans of time. So it had this really slow rate of forgetting. And using this interpretable with respect to time scales, RNN, we then used our usual linear prediction framework to predict the brain activity of different brain regions. But because we had explicitly controlled the time scale at which each unit in the RNN operates, we could then look at simple metrics like feature contribution to identify which parts of the brain relied on which types of timescale units. So what we found, for example, is parts of the brain here, for example, this is your auditory cortex, really relied on the short timescale units for their activity to be predicted. In contrast, this particular part of the brain in your prefrontal cortex really relied on the medium to long timescale units. And by using this simple feature attribution method and an interpretable language model, we were able to come up with this very detailed map of the processing timescale of each brain region. And what you can actually see here is that really nice hierarchy I was showing you in the in silico experiments, but this is much more detailed because it can give you precision up to like a couple of seconds. And you see this very beautiful hierarchy from the yellow regions in your auditory cortex all the way up, and then again from your posterior prefrontal cortex all the way to the front. And so I hope I've demonstrated very quickly what adding interpretability from the ground up can do for you to then interpret something about the brain. Yes?
So before you were using a causal transformer to produce the embedding, then you were using to do linear regression to brain activity, and now you're using an RNN. So these are two completely different architectures.
Yes.
And it seems the representation of these two completely different architectures, they are both equally good to predict brain activity. So this is a bit of contradiction because a lot of people use this ridge regression technique, which they call brain score, to say that actually there is similarities in
Yes, actually there's a catch here. So unfortunately, an RNN is not as good as a transformer. This could be because we just haven't gotten very good at training RNNs at the scale that we're able to train transformers. So this general rule holds that the better your language model, the better your brain prediction in general. There is a plateau, but that is the rule. So this RNN does not predict brain activity as well as the model I was showing you earlier, that your R-square is smaller, but there's an important difference. This model is interpretable. And so you're faced with this trade-off, which I think we just are in neuroscience, wherein you have, do you want the best predictive model or do you want an interpretable model? And they don't always line up. But what I'm trying to demonstrate here is that adding interpretability for a model that is pretty good at brain activity might be more useful than just predicting the brain with something like llama and then not knowing what to do with the internal states of that model.
Yeah, my point was more like that having embeddings that are predictive doesn't imply that the operations that produce those embeddings are similar to the one that produce the representation in the brain.
Right. So
similarities in terms of intermediate representation doesn't establish similarities in terms of like operations.
Oh yeah, so yeah, exactly. So we're not making any operational claims. I'm not saying anything about how the brain is a recurrent neural network or an LSTM or a transformer. It's very hard. That's a very cool question. It's a very important question, but I just don't think we're there yet. At least in the approaches I've taken in my work, you can't really use any of these paradigms to make operational claims. You use the paradigm to make representational claims. The information contained in this representation must be similar to something a particular part of the brain is doing, because you can generate the brain activity of this part of the brain well. So I'm only making representational claims and not operational or algorithmic claims.
Thanks.
Okay, we're a little behind. So let's see. Okay, so in the third part of this talk, I want to now present a different idea of interpretation. Sorry, I'm just throwing these interpretability ideas at you, but I just want to convince you that this is a very powerful paradigm, and there's so many different ways you can look at this problem to understand the brain using language models. So in this particular part of the brain, and actually it connects to the audience question we were just asked, what if you don't have an interpretable language model from the ground up, right? Like, what if there are some types of interpretability you don't know how to build into a model. So in this particular work, I was trying to focus on the parts of a language model that we are confident are interpretable. And in this particular study, I focused on transformers, and I tried to identify parts of the transformer that I was able to interpret well, and then link that to predicting brain activity. So everything I showed you up to this point was functional MRI. It was from my PhD work. Everything from here on is from my postdoc work in the past year. And we're going to now move to a different data set. So here I'm also going to show you that all of these interpretability methods are agnostic to the types of brain data you collect. It doesn't matter what your language model is, what your brain data is, but I'm trying to present this framework of understanding the brain. So in this particular part of the talk, we're going to focus on those single neuron recordings I was showing you with NeuroPixels probes. So here, I modeled the activity of every single neuron that we recorded using a transformer. But specifically, I focused on two parts of the transformer that I know to be interpretable. First is the position encoding. Transformers heavily rely on having position encodings to maintain the order of information in time, to maintain, to understand the sequence in which information came in. And I was just curious to know whether something like a position encoding or something like an explicit temporal code exists in the human brain. Are there single neurons that explicitly represent time independent of what is happening? And in another part, I also did some interpretability work on understanding what the individual attention heads are doing. Now, there's a lot of phenomenal work in both language AI and speech AI where people have found different attention heads of these models to be extremely interpretable in a way that the hidden states are not. So I tried to interpret the different hidden states of a model like Whisper or Ubert, and then I linked that interpretability to how well those attention heads predicted single neurons. In the interest of time, I won't talk about the attention work, but if you're interested, I can tell you what we found. So let's just focus on the position encodings. So again, the grand hypothesis here is, suppose our human participant is listening to a sentence like, "Junior, what on earth's the matter with you?" Are there single neurons in that person's brain that are encoding information like a position encoding? They are responding to specific instances in time. Obviously, I found those neurons, otherwise I wouldn't tell you about this. But what we actually did find was time cells in the human speech cortex. You might have heard of time cells in the hippocampus and play cells and things like that. But for the first time ever, we're finding evidence for single neurons in the human auditory cortex that explicitly capture this representation of time. And the way I found these neurons was by factorizing how much that position encoding adds to predictive power. So what I basically did is I built these separate predictive models using the hidden states of a speech DNN, which we know works really well. We know that the hidden states work phenomenally well. But I also checked how well the position encoding works alone. And what we found was there were a bunch of neurons in the brain who were as well or sometimes better predicted by the position encoding as that very rich content and context-dependent hidden state representation. So let me just show you what the spiking activity of that neuron looks like. On the x-axis is time. This is sentence onset. This is the offset of 100 different sentences on the y-axis. And this is the spiking activity of that neuron. Now, even if you've never seen a neuron fire before, I'm sure you can appreciate that this neuron loves the beginning of speech. So as soon as you go from silence to speech, this neuron gets very excited and responds independent of what the actual sentence is. It doesn't really care about the content. It cares about time. It cares about speech onsets. This is another neuron that really loves the middle time point. It really responds around 1 second in. And this is a third neuron that responds towards the end of the sentence. Now, obviously, these neurons don't exactly know when somebody's going to stop speaking, right? Like, you can have some prediction, but these neurons don't exactly know when the sentence is going to end. So as you go farther and farther in time, the temporal receptive fields of these neurons becomes broader and broader. So it goes from being a very clear line to slightly spread out and quite spread out. But what I'm trying to show you here is that there are these neurons that respond to different sections of time explicitly, independent of what the person is hearing. And let me just show you what this looks like at the population level. Again, on the x-axis is time from the onset of sentence to about 1.2 seconds in. On the y-axis are about 35 different neurons. And the order of the neurons are the same across the 3 plots. And these are 3 different sentences that our human participant heard. Bright pink implies that that neuron really fired at that particular time point relative to everywhere else. And what you actually might notice is that these plots are very consistent. So let's take this neuron, for example, neuron 25. Independent of what sentence the person was listening to, this neuron always fires around 600 milliseconds. And so you see this across the entire neuronal population. You see this tiling of time from the beginning of speech to a second or more in. And so you have these explicit time neurons in the human speech cortex. And we were only able to figure this out by using these explicit position encodings that have no content information, just time information, and evaluating whether that time information is sufficient to predict the activity of these neurons well. Okay, now the final part of my talk, I actually already told you all of the details for this, and I'm not showing you any results because this is very much an ongoing project, but I'm very excited about it, and I just wanted to present this idea. But in everything that I've showed you up till now has a lot of different ways to think about interpretability, but it's all in this constrained framework of linearized models. So you need to really think a lot about where you're going to add interpretability in the model, and then how you're going to use that linear projection from the language model to the brain to understand something. But you can also just take a cop-out and have the model do the work for you. So you can first take your massive neural data sets. It could be neuropixels, ECOG, fMRI. It could be all of these together. You can take a lot of different types of language data. For example, somebody listening, somebody speaking, reading, writing, many, many different languages. And you can simply directly feed it into an artificial network. So instead of extracting features that may or may not be interpretable and doing a linear projection, you simply take your language stimulus, feed it into this artificial network, and make it predict brain activity directly. And the reason I think this is a very interesting and powerful approach is then you can use all of the AI interpretability tools that the field has come up with to understand the internal dynamics, the representational geometry, the types of features that this model learns as a clue to something about what the brain could be doing. And I And a fancy word for this would be that this could be like a silicon brain. If this model works really well and has great generalization across people, across cognitive tasks, you can really treat this as a silicon model of a human brain when it's processing language, or a foundational model of language processing in the human brain. And again, this is early work, but we're able to see very good performance with these models. These models are really good. They have great zero-shot performance. And we're still working out the exact details about architecture and stuff like that. But I do think that this is the future, especially if you want to leverage patterns in vast data sets like AI has done. I really think it's worth trying to build these artificial networks that are trained directly on brain data. So if you're at UC Berkeley or UCSF and you're interested in our research, please get in touch. I would love to chat about this, and I would love to collaborate. So that is it. Through my talk, I hope I've convinced you that this is not a lost cause, and even if you use something that is fundamentally uninterpretable like a language model, you can understand something about the human brain through these 4 different ideas. So yeah, I would like to thank all of you for your attention. I would especially like to thank my PhD advisor, Alex Huth, and my postdoc advisor, Eddie Chang at UC San Francisco, and all of the mentees that I've had the fortune of working with and contributed to various aspects of the work I was showing you. Also my fellowship agencies throughout my PhD, and the 2 different societies that have appreciated my PhD work, and finally the industry collaborators that we've worked with. So thank you.
Great. Are there any questions?
This makes me think. So, first of all, great talk. A ton of cool stuff going on. It makes me think of Laura's work, where she had sort of like the cascading in time of the phonemes, like how over time different neural assemblies were representing that information.
Yeah.
Do you see like a parallel to your time work?
Yes, yeah. So I think what Laura's work has done is it's done a really great job at identifying the idea of time scales at a sublexical level. So I think a lot of her work has focused on these very fine time scales, especially with the neuro recording modalities that she works with that have good temporal resolution. And I think my work is complementary to that in thinking about time either after the sublexical level, like once you're at words, once you're at phrases, what that time could mean for things like semantics, or at a much lower level than she's operating at, which is in these single neurons, which probably then work together to arrive at phoneme categories. So I think between us, I'm hoping there is an end-to-end timescale theory.
Yeah, very cool.
I can understand why it's important to find the beginning of a sentence, for example, to find structure.
Yeah.
But why is it useful to have a neuron that fires after exactly 600 milliseconds? What does it do for the understanding of processing?
Yeah, great question, and something I've wondered about too. Like, so fundamentally, we know that time is important for speech processing. Like, if I say "k at," that's a different word than if I said "t at k." The sounds are the same. There's some co-articulation differences, but the order of information is different. And so the brain must and definitely has some mechanism of not only keeping track of what happened, but also when it happened. Now, there are many theoretical possibilities for what that could look like at a single neuron or population level. The explicit time representation is just one of those possibilities. Like, you could have explicit time neurons and neurons that only care about what happened, so what and when, and then these neurons maybe feed into a higher level, either a region or population or another single neuron that then combines that what and when to learn this content plus time representation. And so I didn't show this here, but we also find evidence for neurons like that. Like, there are these pure time neurons, but there are also neurons that respond to specific phonetic categories, but only when they happen at a specific time. Like, they love vowels, but only vowels at the beginning of a sentence or in the middle of the sentence. So we're finding evidence for pure time neurons and time plus content neurons, and then neurons of a third category, which are much more contextual. So they don't care about just phonemes or just time. Like, the third category is neurons that care about what happened in the context of what happened before, independent of when it happened. So they care about like phonetic sequences independent of when those phonetic sequences occurred. So we're finding these many different types of computational motifs, and I don't have a clear story, but what I'm hoping to work out in the future is understanding how these neurons could be talking to each other to ultimately give rise to speech perception. Like, how do they communicate with each other so that we're able to keep track of both time and content?
Yeah, great talk. Yeah, I was a little bit confused by the fact that you said that all these meanings or equivalent things for particular concepts were localized.
Yeah.
But on the other hand, I mean, it's also well known that the activity is quite distributed because there are like associated memories that fire and things. So how do they?
How do these two fit together?
Yeah.
Yeah. So, like fundamentally, what we find is if you just look at an fMRI map of somebody listening or speaking, listening, a lot of their brain lights up. A lot of their brain reliably lights up. So we know that language and any downstream aspect of language is a wide distributed phenomena. But even if that is true, which it is, you can still find important functional differences. So what I'm trying to do here is we know all of the regions that are colored are activated when language happens, but what are the functional profiles of those regions? Like, how can I be sure that this part will light up for a specific language stimulus? And by identifying the semantic selectivity and the time scale of that region, I can develop some sort of a theory about what drives that region and which type of network it might be playing a role in. So things like semantics and syntax is very confusing and everybody has their own idea, but things like semantics and syntax and phonemic processing might happen in specific regions, but altogether all of these regions are working together to give rise to our perceptual experience of language.
Thank you.
I guess, great talk. I guess one of the interesting open questions about how the brain is organized is around the question of how localized is the information coded in different places. And it strikes me that with the kind of modeling you're doing, you could, for example, look at data from a probe and also from an fMRI, maybe the same person at a different time.
Yeah.
And you could then ask the question, what fraction of the whole brain activity is actually encoded in this particular local probe location? And that and then virtually you could ask that question in your virtual brain in many different locations.
Oh yeah, that's right. Yeah.
And it might be an interest. Have you thought about that or tried any of those kinds of
ideas? Yeah, no, actually that's a great idea. And it would actually be very interesting. So one thing that I haven't yet done is combine these different data sources to try to build a more cohesive theory about how all of this works together. So I've done some very preliminary analyses with the NeuroPixels work, and what we didn't have fMRI, but for a participant whose neuropixel activity we were recording, we were also able to capture the population activity of the ECOG electrode on top. So you have the population activity of the electrode on top, and then you have the single neuron activity across the cortical column. And Laura and Matt Leonard did some preliminary work on this, and I developed on that further. And what we actually find is single neurons can have very, very different types of functional properties that may not always relate to what you're capturing at the global level. Which is obvious because you're subsampling with NeuroPixels, but by just looking at the bias of the neural population, you can guess what the ECOG activity could be. So for example, if many of your ECOG electrodes tend to be context sensitive up to 500 milliseconds, you can be assured that the ECOG electrode sitting on top cares about long time scale information as compared to single neurons that care about just the current time representation, and then the ECOG activity on top cares about very specific instances of time. So I think it's a great idea and I would love to explore further, but we've done very basic work in trying to link ECOG and neuropixel activity. This is not my work, but Ev and others at MGH have done work on trying to understand how semantics could look like at the single neuron level. And what I took away from that work is that semantics doesn't really, you can get single neurons that are tuned to one concept or the other, but something like this is a much more population level code. So something complex like semantics needs a lot of neurons and a lot of different pieces coordinating with each other. So to add to your question, I think there's also this interesting question of what types of linguistic phenomena require specific spatial scales of representation. And one thing that I'm interested in is, can you ever study something that you can study with fMRI or ECOG at the single neuron level? Does it even make sense to think about something that is a global property or a much more distributed property at the single neuron level? And I think that's a very interesting question that we should figure out.
Yeah, super interesting talk. I was sort of interested about your silicon brain next steps. And this might be maybe a silly question, but so the idea is you want to no longer hard code these features, you would like to just give directly the speech and then see if you can recreate the brain signals from that. But what I'm wondering is, we have a lot of mechanisms in our body that encode this sound or vision or something into a very specific way. So how do you feel that this encoding, like this way that sound is encoded via microphone, how well can it relate to this sort of thing that we have in our ear? Is it similar enough, or is this an issue at all?
Yeah, good question. So I think fortunately, and maybe people in the audience know more about this than I do, but fortunately for things like our cochlear system, we now have very good computational models of how that works. So things like cochlear representations, for example, we can be confident that we can develop a computational model for that. But everything after that, as soon as you enter the cerebral cortex, it's a mess. And so I think that's where something like this could be very interesting. So one way to sort of add interpretability or an implicit bias in your foundation model would be to start with the cochlear model and then feed it into something like this, because like you said, we know that there is this specific pathway that the brain follows. And similarly, we could do something like that for vision as well. So I think the reason I think this is an exciting opportunity is because you can bake in the types of constraints we know exist in the brain, and then you can evaluate what the model learns with those constraints in place.
What would be your architecture of choice for that neural network that map the speech into the brain activity?
Yeah, I think that's a hard question because A, it's empirical. It could be that the architecture, you get the best architecture for a particular participant is not doesn't generalize well for another. Could be that the best architecture for a specific cognitive task doesn't work for another. For example, language foundation models might look very different from vision foundation models. We just don't know. So I think it's an empirical question. And I'm a little skeptical, but I would love to hear everybody's thoughts about making mechanistic claims based on the choice of architecture. Like suppose we found that transformers work really well, I think we would have to think a lot about whether that is important and whether the transformer being very good tells us something about how the brain could work at a mechanistic level. So I think architecture is a very hard question, while representation I think is easier. But yeah, so I just think it's empirical.
Yeah. Thank you.
Yeah, I just want to follow up with one comment about the brain has many activities versus a very strong localized signal. Yeah. So my background is in statistics. Actually, in statistics, in linear model, usually in standard linear model class, we learn, okay, like you can identify important variable that can explain the variability, the R square. But I think modern statistics are also thinking a lot of weak variable, like a lot of variables that have very weak signals. They combine have a very strong relevancy, and our ability to detect them is increased by the sample size. So I feel there is a possibility in your model that you can maybe employ more modern statistic technique to find actually many weak signals. Variables, which in your case is some other area of the brain.
Yeah, I think that's a great point, great suggestion. So one thing that I shoved under the carpet was for a lot of these approaches, like you said, you're very limited by the statistics of like the feature space, for example, or the stimulus that you use. So just as a very naive example, and to follow up on your thought, if your language stimulus never has any information about cats, it's very hard for you to find a part of the brain that likes cats, right? Like the statistics of your stimuli, the statistics of the representations of the language model, all of these are adding many, many different levels of biases in the types of functional properties you're estimating. And of course, there's another added complexity that if you collected more data, do your results generalize? Because now you have the power to estimate things which initially had smaller effect sizes. And we discussed this a lot in my this opinion piece we put out, the second one, about this in silico paradigm. And my current thinking on this is, because this is such a tricky problem and biases can come up in so many places, the best thing to do is verify your results in all ways possible. So suppose I estimate some functional properties using my language fMRI data set. Now, if I used your fMRI data set, which had different participants, different stimuli, do you still come up with these gross patterns? Or if you used, instead of ChatGPT or GPT, if you used I don't know, Claude, do you get the same? You get my idea. Like, if you substitute out different parts of your modeling framework and you still come up with the same answer, I think that is what we need to make to be confident that our estimates of the brain are correct.
Great. I think we're running out of time. So thanks so much for a great talk. We'll give you a break, and then we'll speak at 11. Thanks.